Good afternoon. Hey. Thank you for the nice introduction. I hardly have to say anything after that, but I will. So I was asked to speak to you today on this occasion, the campus on the Common, as we celebrate the 50th anniversary of the University of Massachusetts, Boston, to tell you a little bit about the new Center for Personalized Cancer Therapy. But to do that in the best possible way, let's step back to see where the center came from so we can see where it's heading. Okay. So this is a little history lesson. Back in the day, this would be about 25 years ago, when I was a graduate student. There is gray hair under here, it's just cleverly disguised. The current thinking was that cancer was cancer. No matter what tissue or organ harbored them, cancers were all alike. And they all could be killed with a magic bullet, some kind of chemotherapy that awaited to be discovered. But we all believed it was there. This turned out to be true in only a limited number of cases, and I'll tell you a couple of those. Testicular cancer is one of those cases. In 1970, 90% of patients diagnosed with testicular cancer died of their disease. Since then, the use of chemotherapeutic agents like cisplatin in particular, plus surgery, has raised the survival rate to 80%. So we went from 90% mortality to 80% survival. We had a magic bullet. It was called cisplatin. It really worked. Pediatric leukemias are another example, especially the common acute lymphoblastic leukemia called ALL, common childhood leukemia. These ALL patients universally died of their disease just 50 years ago. So 50 years ago, just like 50 years we're celebrating when UMass Boston was formed, if a child had ALL, it was a fatal diagnosis. Today, because of advances in developing effective chemotherapies, magic bullets, 90% of children diagnosed with ALL survive their disease. So we went from almost complete mortality to 90% survival. So these are the success stories. Okay, this is the good news. But you know, for most cancers, that's not the case. So why is that? This is largely because our prior understanding of cancer was as a homogeneous disease. No matter where the cancer occurred in the body, all these cancers were the same. That was incorrect and was actually very simplistic, although we didn't know it at the time. So here's an example of what I'm talking about and how we can kind of visualize this. If everybody could, hold up your hands. This is an interactive discussion, okay? Hold up your hands, okay? So what are these things at the ends of your hands? These are fingers, right? So we identify these things as fingers. You can put your hands down now, okay? They all pretty much look alike. They all belong to hands, which also pretty much look alike. Okay, this is how we used to think about cancerous tumors. They all looked alike. They all functioned alike. So if we could find a magic bullet that would kill one tumor, it would be able to kill them all. That was wishful thinking. All right. We now know that all cancerous tumors are not alike. A breast tumor is not the same as a prostate tumor. A prostate tumor is not the same as a lung tumor, and so on. So let's hold up our hands again, okay? I told you this is interactive. All right. So take another look at your fingers, okay? When you really look at your fingers, you realize that actually you've got 5 different ones on each hand, right? You got a thumb, you got a index finger, a middle finger, a ring finger, and a little finger, okay? So even though these are all fingers, they are all different types of fingers, okay? So you can think of cancerous tumors in the same way. Breast and prostate tumors are different from each other, just like a thumb and an index finger is different from each other, okay? Fingers, but not the same. So cancerous tumors, similar in some ways, very different in others. Because even though breast and prostate tumors are both classified as cancers, their characteristics depend to some measure on which organ or tissue gave rise to that cancer. These differences are due in turn to the genetic programming that makes a breast, a prostate, or a lung. So just like the organs are different from each other, so the tumors that are harbored in those organs are different from each other. Okay, we know that now. We didn't know that 30 years ago. So that message actually took a long time to sink in. And it wasn't really until the 1980s that scientists and physicians figured this out. Okay, so that was progress. But we still didn't have the whole picture. We also used to think that each type of tumor was relatively homogeneous within the group. So a breast tumor was a breast tumor, and all breast tumors were the same. However, that turned out not to be correct either. If you had 10 people in a room with breast cancer, they were all medically managed in the same way just 20 or 30 years ago. It became apparent that one size did not fit all in terms of breast cancer therapy. Some breast cancer patients would fully respond to surgical removal of the tumor. They'd never have a recurrence. They were fine. Other patients who got the same treatment suffered a recurrence. Their tumors would come back, and often they'd be metastatic, and the patient would have a very poor prognosis. So clearly, even though 10 patients might be diagnosed with what looked to be very similar types of breast tumors, the tumors themselves would respond differently to therapy. So also clearly, these 10 breast tumors were not really the same. Okay? So let's try to visualize this. Let's hold up our hands again. And this time, you only want to hold up your index fingers, okay? So look around the tent and look at all these index fingers. You can see very clearly these are all index fingers. But my index fingers don't look the same as yours or yours or yours, okay? So even though they're all recognizable as the same thing, they are clearly not identical to each other. All right, so you can actually take that analogy now and apply it to tumor types. So now we know that although all breast tumors are recognizable as breast tumors, there are clearly differences between breast tumors in different patients. And this makes a difference then in how to medically treat those tumors. This makes the job of curing cancer all that much harder, which is why this concept was really a bit hard to swallow among those of us who are trying to cure cancer, okay? It was kind of like a bitter pill, actually. We didn't want to hear this. But we're now recognizing that we must actually defeat cancer on a first-name basis, hence the title of my talk. We have to recognize that each breast cancer is as individual as the patient in which it resides, and that each cancer has to be treated on an individual basis. If we really want to affect cure, that's how we have to approach this. So this is the basis of personalized medicine and personalized cancer therapy, and that's really why the Center for Personalized Cancer Therapy exists, okay? So that's all the backstory. So now let's hear the forward story, right? So there's actually 3 major ways that the CPCT, which is our abbreviation, is rising to the challenge of defeating cancer on a first-name basis. First, the CPCT is developing methods for tumor subtyping. We are asking what makes one breast tumor or prostate tumor different from another. Can we develop ways to identify those differences up front so that clinicians can make use of that information and tailor treatment strategies specific for individual tumors? Okay. I won't go into a great deal of detail here, but basically the CPCT is developing tools that will permit tumor subtyping, and that would have great clinical utility for physicians treating cancer patients. Okay. Second, the CPCT is developing tools that can predict a tumor's response to therapy prior to the more conventional imaging methods that we use today. So currently, when a patient goes on chemotherapy, it can take months before any changes in tumor size are detectable by MRI or CAT scan or PET scan or any of the other kind of medical imaging modalities. That's a long time in a cancer patient's life, to wait to see if your chemotherapy is working. Okay, and the one thing cancer patients don't have is time. So if the tumor is found to be smaller after 3 months of chemo, that's a great thing. Okay, the patient's on the road to recovery. But if the tumor is not found to be smaller, so it's not responding to the therapy, or if it's actually bigger after 3 months of chemo, then the patient has lost time and the tumor has actually progressed. And you can't get that time back. So the patient will then have to go on a different, maybe experimental chemotherapy, and is that much further away from being able to recover from their illness. So we need tools that can tell a physician earlier than 2 or 3 months whether a patient's tumor is actually responding to the chemotherapy. That way, if the tumor isn't responding, the patient can be switched to another therapy that might be more effective. And so we're developing tools to help physicians see whether or not the chemo is working fairly immediately after the patient starts chemo. Okay. Third, the CPCT is training future cancer researchers, nurses, and clinicians. We literally need more hands involved in cancer research and treatment. So at UMass Boston, we're training the next generation of cancer researchers, as Dean Jackson mentioned, who will be able to pick up the baton and run the next relay in the race against cancer. We are training nurses who will specialize in oncology and help cancer patients through the process of cancer diagnosis, treatment, and hopefully recovery from cancer. We are training students who will go on to medical school to become surgical, radiation, or medical oncologists. These are the doctors who treat cancers, and so they will learn to effectively treat cancer patients. As a center within an educational institution, excuse me, swallowed something wrong there, the training of future scientists, nurses, and physicians, thank you, is focused on beating cancer, and this is a primary element of our overall mission. So that, in a nutshell, is what the Center for Personalized Cancer Therapy is all about. Thank you for your attention. I hope I helped increase your understanding of cancer research and treatment and of the CPCT. I also hope that you don't suddenly look at your fingers in a strange way after this talk. They're perfectly normal, okay? They're not cancer. They're not tumors. They're okay. If you'd like more information, you can Google CPCT, and we're actually the first hit. So don't ask me how that happened, but it did. And you'll get our website, and I can answer questions now, and I'll be sticking around a little bit afterwards as well. So thank you very much.
I think you can understand why the Chancellor and the Provost were so thrilled when Jill accepted their offer to join UMass Boston from the University of Michigan Law School, where she was professor
No,
of
medical school.
Medical school. I'm sorry. I've been under the tent, and it's.
Yeah, I got another degree.
Yeah,
you know?
No. Professor of urology and cellular and molecular biology, cancer biology, and computational medicine and bioinformatics at the University of Michigan School of Medicine. So, what a coup that we have Jill here at UMass Boston. So,
thank you.
Floor is yours. Questions about our personal vulnerability and opportunity for the kind of cancer treatment that she's pioneering. Yes, please.
It's about therapy, and you mentioned about that once like a cancer is switched to another part and they have different kind of therapy. It's like, so what about like, is that because their white blood cell is down, or is one cancer causing another cancer, so they need to switch to therapy?
Oh, so there's a lot of reasons why a tumor may or may not respond to a particular therapy. Some of it is the real basic biology of the tumor, where it's driven by different mechanisms in one tumor than in another. So if the drug is specific to hit a certain target in one tumor, and that target is the reason why that tumor is a tumor, then the tumor will respond, okay? But if it's not the right target, then the tumor won't respond, and it'll actually progress. So there are certain pathways that we know of in tumors that seem to be the most important. These involve different proteins in the cell that are acting up, if you want to put it that way. They're activated when they're not supposed to be. They're being produced when they're not supposed to be. And so if we target the right ones that are actually making the tumor grow and advance, then we've hit an effective cure. But if not, we haven't. And right now, there's very little stratification amongst tumors. It's not that there's none, okay? There is some. So the 80s was kind of a turning point. That's when a professor by the name, he's actually an MD, he was a medical oncologist, Dennis Slayman, found that in a lot of aggressive breast tumors, okay, the ones that were not responding to therapy, there was a certain protein called HER2 that was very highly expressed. And it took 10 years, but finally, there was an antibody or a therapy directed against HER2 called Herceptin. You might have heard of Herceptin, okay? that actually came out of that lab showing that this protein was really way up there, highly expressed, and it was actually causing the cancer to grow and metastasize. And so the idea was, let's hit that target, hit that receptor with Herceptin. And actually that helped. That helped a lot of women who had aggressive breast cancers. It prevented them from going on to metastasize. Okay? So once you know the biology of the tumor, and not all breast tumors have this problem, but some did, and that's why they weren't responding to conventional therapy. So once you know what's driving the tumor, then you can develop therapeutics to hit those actual targets. Okay? So one thing we're doing in the CPCT, it's not so much that we're developing therapy, we're developing more the tools that clinicians need to figure out what a tumor is doing, okay? Whether it's this tumor A type, tumor B type, or tumor C type. So right up front, they can say, okay, this tumor we know from the markers that we got from the CPCT, this tumor's not going to respond to, say, doxorubicin, okay? So we're not even going to bother to give the patient that, they're not going to respond. We're just going to put them on hormone ablation therapy because they have those markers, okay? So we make the therapeutic choices more appropriate up front. That's really important. And then we are developing tools so the physicians can track whether the tumor is responding, okay? So rather than waiting, like I was saying, months to see on an MRI that the tumor's gotten bigger or smaller, you should be able to tell sooner that the therapy is hitting the mark and the tumor's responding. And so we're making biomarkers to show that. And so that will save a lot of time in misdiagnosis and mistreatment, which is unintentional. You know, doctors are doing what they can do with the tools that they have, but they need more tools. That's the important thing. Okay. Mm-hmm.
Hi, I completely understand the clinical and therapeutic foundation that you're describing of the center, but I'm curious to what extent, as your title indicates, one-by-one personal treatment, whether the center also is involved with patient-reported outcomes measures and the coming growth of that modality.
Yeah, so that's actually kind of a problem. So patient-reported outcomes are not super quantitative, okay? And that's an issue. So patients can report whether their own symptoms are abating, getting worse, whether there's, you know, let's put it this way, they can fill out surveys and questionnaires. And it's not that that data is not useful, it is, but it's not good enough. So there's more and more of an attempt to do what we call phenotyping, which is really using measures that are really quantitative. And that's more at the molecular level. So we're getting away from kind of the self-reported stuff and getting more molecular about it.
One last question? If not, please join me in thanking Professor Macoska for her presentation.